Now we'll complete the study guide for the cells unit exam. We're picking up at number 19. It says, if a red blood cell is placed in a concentrated salt water solution, it would, concentrated salt water, that would be a hypertonic solution. It has more salt than the cell. So the cell would lose water and shrink. So it would shrink. Number 20. If a red blood cell is placed in a 0.9% salt solution and does not gain or lose water, the solution is, it's not changing, it's not gaining or losing water. And we've seen several times that the red blood cell is 0.9%. That would be isotonic, iso meaning equal or the same. So this would be an isotonic solution. The cell does not shrink or swell. There is no net movement of water. Number 21, water leaving the tubules of the kidney in response to a hypertonic fluid surrounding the tubules is an example of anything that says water moving. If water is moving, then it is osmosis. And this is an example of osmosis. Number 22, when there is a lower concentration of water outside the plant cell, rather than inside the plant cell, the plant will tend to lose water and shrivel, wilt, shrink. Being a plant, we're going to say wilt. That you may put the cells will shrink. And they're losing water. Number 23, a biology student placed some apple pieces in distilled water and some in salty water. The cells in the apple pieces will blank in the water, blank water in the distilled water, and blank water in the salty water. Well, distilled water has a lot fewer particles in it than the apple pieces, so water is going to move toward the apple pieces and they will swell. Swell, and they also become rigid. And in the distilled water, is a hypotonic solution, so the water is going to move into the cells and it's going to swell or become rigid. The salty water has more salt, more solute particles than the apple. So in the salty water, it would be a hypertonic solution. So water is going to go out of the cell, and the cell is going to shrink and become floppy. Become limp is what we put. Shrink, and that would cause the apple piece to become limp. Osmosis is an example of simple diffusion. It doesn't require energy, doesn't require a protein gate, so it's just simple diffusion. If placed in a hypertonic solution, the plant cell will hyper has more salt, so the cell will shrink. Number 26, an animal cell that is surrounded by fresh water will burst because the osmotic pressure causes water to move into the cell, water to move out of the cell, solutes to move into the cell. Well, solutes are not going to be moving, so we can automatically eliminate those two. Water's what's moving, and it says it's in fresh water, so that's a hypotonic. Water is going to move into the cell and cause the cell to swell and possibly burst. So water is going to move into the cell. If it's talking about solutes moving, we can automatically eliminate those answers. Okay. And it says it'll burst, so if it's going to burst, it would have to be swelling, so water would be moving into the cell. So read those questions and use your test-taking skills carefully. What would you expect to happen if you placed a typical cell in water? Okay, if it's in plain water, we would expect water to move into the cell, because there are particles inside, the solute particles inside the cell. So there's higher concentration inside the cell, so water is going to move into the cell, causing it to swell. What would happen to a saltwater fish in fresh water? And if you need to draw a picture that you can do that, you would have your little fish in your salt water. Well, your fish is fresh water, so it's going to have a low concentration of salt compared, no, your fish is saltwater, sorry. It's a saltwater fish. So your fish is salt water. Highlight that because that's our keyword right there to help us remember. Saltwater fish, and we're going to place it into fresh water. So saltwater fish would have a lot more solute particles in it than the fresh water. So our fish, let me put that down, our fish is hypertonic compared to the water. And the water is hypotonic compared to the fish. So the fish is in a hypotonic solution, a hypotonic environment. So we can put, therefore, water will move into the fish causing it to swell, and if not rescued, die. Polar versus nonpolar. Okay, plasma membrane. Remember, it's a lipid bilayer. These are phosphate heads. Phosphate heads like the water. The lipid tails, remember, lipids are fats, and fats do not dissolve in water. They are hydrophobic, do not like water. So water is going to be close to these phosphate heads because they do like the water. So where would you most likely find water in the picture? You're going to find it close to those polar heads. Water is a polar molecule. It's attracted to other polar molecules. So the water is going to be close to the polar heads. The endosymbiotic theory. The endosymbiotic theory is an attempt to explain the presence of chloroplast and mitochondria in eukaryotic cells. Chloroplast and mitochondria. So the endosymbiotic theory is talking about the chloroplast and the mitochondria. So in evolution of eukaryotes, cells that contain mitochondrion-like organelles. So mitochondria is your key word there. Containing mitochondria-like organelles have an advantage because they can make more use of available, if it's mitochondria, mitochondria is going to give us what? Mitochondria is going to give energy in the form of ATP. So if it's got a lot of mitochondria-like organelles, then it's going to have more energy in the form of ATP. Mitochondria and chloroplasts reproduce, well, they reproduce like bacteria, which is binary fission. They just kind of split into and make identical copies. Binary fission. Same way that prokaryotes reproduce. Homeostasis, there's that big word again. Homeostasis, we talked about it probably like the second week of school, and I said you'll probably see that word pretty much every day for the rest of the semester. Well, here it is again. So what is homeostasis? Okay, homeostasis is maintaining constant internal conditions suitable for life even when the external conditions are changing. That means we go outside and it's 30 degrees, our body temperature doesn't drop to 30 degrees, it stays around 98.6. That means we go outside and it's 100 degrees, our temperature doesn't automatically go up to 100, it tries to stay around 98.6. It means our blood pressure stays just below 120/80. Our blood pH our body has a way of keeping our blood pH around 7.1. That's all part of us maintaining homeostasis so we can be healthy and carry out life functions. Because if we're out of homeostasis, then we're not going to be healthy and able to do the things our body needs to do. So homeostasis is maintaining those constant internal conditions. Number 33, which term describes the relatively constant internal physical conditions of an organism? Well, there it is again. That is homeostasis, maintaining those internal conditions. Homeostasis. So Venn diagrams of similarities and differences between endocytosis and exocytosis. Okay, so some similarities, both are bringing, I'm just going to list them since, well, I could draw a Venn diagram, but anyway, we'll just list it for right now. Similarities. And differences. Okay, so similarities would be, similarities, both are taking, similarities between endocytosis and exocytosis. Both are transporting molecules into or out of the cell. We'll just put both are transporting molecules. Differences, endocytosis is moving in. Endo sounds like in, so endocytosis is moving in. Taking particles in. Exocytosis sounds like exit. Exit. So if things are leaving the cell, it's exocytosis. So exocytosis is moving particles out of the cell. Another similarity would be both require energy. They're both active transport. Require energy. Both are transporting molecules, both are active transport and require energy. Endocytosis is taking particles in, and exocytosis is taking particles out. I hope that helps. Have a good day.